Dear students, Assalamu alaikum. Today we are going to talk about a series of lectures. These lectures will be part of our course of special topics in biochemistry, course code CHEM 6462. The title of this series of lectures is Contemporary Biochemistry and Molecular Biology Techniques. So let's go ahead with the lecture. The outline of this lecture is polymerase chain reaction. Then we will go and talk about gel electrophoresis. After this we will talk about quantitative real-time PCR or QRT-PCR. This will be followed by immunohistochemistry and immunocytochemistry. The reason for selection of these topics in this sequence is because these topics or this series of lectures they will help you to understand these topics in a sequential manner. So today we begin with the first part of this series of lectures, and that is polymerase chain reaction or PCR. Polymerase chain reaction provides an extremely sensitive mean of amplifying relatively large quantity of DNA. This technique was first described in 1985 by Carey-Bolus. He got Nobel Prize in 1993 for this very important invention. Since polymerase chain reaction was introduced into the field of biochemistry and molecular biology, since then it has got enormous importance, and with the passage of time it is getting increasingly important with new techniques and new methodologies that are being introduced in this field. Materials that we need for conducting a PCR reaction, they include template DNA. By template DNA we mean the DNA that is to be amplified. Then we require DNA nucleotides. DNA nucleotides are obviously the building blocks for new DNA. Similarly, like bricks are the building blocks of say a room or a building, these DNA nucleotides are the building blocks for formation of new DNA. Then we require primers. Primers are single-stranded DNA, and they are between the length of 20 to 50 nucleotides long. These primers are very important for conducting a PCR, because this single-stranded DNA between the length of 20 to 50 nucleotides, they can anneal with the denatured DNA. When the two strands are separated, they have the ability to anneal with them, and then the DNA polymerase enzyme, which is also very important for conducting PCR, it can combine with the it can recruit the building blocks or the DNA nucleotide that are freely roaming around in the reaction mixture, and this enzyme work in the synthesis of new DNA. Then a very important factor is buffers that provide an environment for conducting this type of reaction. And magnesium ion important is a cofactor for the proper functioning of the enzyme system. So there are three major steps in the process of PCR. One of those steps, important steps, is denaturation. Another one is annealing. This is followed by extension, and after extension, when once the extension is complete, the cycle repeats itself from extension to denaturation to annealing to extension. Now in the following slides you will see how it works. So now this is the cartoonic representation of how the polymerase chain reaction or PCR works. This is a DNA, a template DNA. Now when we put the mixture into the in thermocycler, it in the first step it will change the temperature in around 1 minute time at 294 degrees C, which will result in denaturation of the DNA. As you can see, the hydrogen bonds between the two strands, they are weak bonds, they are broken, the two strands are separated from each other. After separation of the two strands from each other, now the thermocycler will bring down the temperature from 94 degrees centigrade to 54 degrees centigrade, and this it will remain like this for 45 seconds. Now this will give opportunity to the single-stranded DNA molecules which are 20 to 50 nucleotides in length to attach with the complementary sequences in the DNA, both in the form of forward and reverse primers. These primers, they will anneal with their respective complementary part. Once they get annealed, the thermocycler will change the temperature from 54 degrees C to 72 degrees C. Now why 72 degrees C? Because this is the optimum temperature at which the polymerase enzyme functions best, and it start to recruit the freely available nucleotide, and extension of the respective strands happens. This happens for around 2 minutes, which is followed by when once the cycle is completed, this extension phase finish, the thermocycler will change again the temperature to 94 degrees C and will cause a denaturation, which will result in again the separation of the two strands. Again it will bring down the temperature to 54 degrees C for 45 seconds. Again the other freely available primers will attach with the complementary part of in the parent single strand of the DNA. Again the process of extension will take place, and this continue for about 30 to 40 cycles of 3 steps. In the next slide you can see through this animation how it functions. You can see that DNA was first denatured like this. Now you can see the denaturation, and then now the primer anneal the DNA polymerase enzyme, which is shown by this yellow. It is recruiting nucleotides. Again you see denaturation of the DNA, again annealing, again extension through the DNA polymerase by the yellow. It happens in both the strands through reverse and forward primers. And you can see different molecules of the RNA polymerase, they function through this procedure. So basically we are seeing 3 steps like I mentioned earlier. That one of the step is denaturation, which happens at 94 degrees centigrade. The other one is annealing, which happens at 54 degrees centigrade, and the other one is extension, which happens at 72 degrees centigrade. So this happens for about 30 to 40 cycles. Let me recap the story. This is template DNA. In the first cycle, the template DNA is separated, and then it is duplicated. In the next phase, this DNA also get duplicated, and so on. You can see that the DNA is exponentially being amplified. You see exponential amplification. So as a result of in 35th cycle, in result of 36 cycles, we see that 68 billion copies of the DNA are produced. One can see a huge number of DNA that are produced as a result of this process of polymerase chain reaction, or PCR. So the simplification is enormous in quantity. Now after this, once this reaction is completed, this process of amplification, we need to confirm the reaction through the process of gel electrophoresis. This is an example of gel electrophoresis reaction. What we see here is ladder. Ladder is a known DNA, and that is run parallelly with the unknown samples to get an idea about the size of the amplified DNA. So these are the different unknown samples, so they can be compared, their size can be compared with the help of this ladder. And we can also, it also tells us about the quality of the PCR reaction of the gel that is being run. So it is another topic. I will talk about this in detail in the coming lectures, but this is just to tell you that PCR reactions are followed by gel electrophoresis to confirm whether we have successfully amplified the required DNA or not. So after this, now there is a very important point that we need to understand, and that is regarding primer designing. This is a very technically important aspect of running a successful PCR reaction. So there are some general notes on the primer design in conducting a PCR that I would like to talk about, because this process of designing a primer plays a very important role in the success of any PCR reaction. So this is why it is said that perhaps it is the most critical parameter for successful PCR in the design of primer. There are certain very important critical variables that are important in running a successful PCR. Is specificity of the primer, primer length, complementary primer sequences, GC content, 3' end sequence, and melting temperature. These are some of the most important parameters that needs to be considered for running a successful PCR reaction. Now, talking about the specificity. Specificity is important because this helps the primer to attach to a location that is specified for it. If the primer attaches to some unspecified location, it can cause a problem. So attachment of primer to its specific position is very important. Primer specificity is at least partly dependent on primer length. So this is why primer length has to be very carefully selected, because it has a very important role in the primer specificity. The chances of 15 base pair oligonucleotide attached randomly is more compared to a 25 base pair oligonucleotide. So now this is very important to understand. if we keep the size of the primer very low, what it can do is that it can possibly do unspecific binding, which can result in a reaction that is not desirable, or because when it binds to the unspecific location, it will amplify a part of DNA that we are not looking for. So we have to be very careful in designing a primer, because designing too short oligonucleotides or primers can be risky. So we have to design primers very carefully. Now, another important point is primer length. This is somehow related with the specificity. Specificity and temperature of annealing are at least partly dependent on primer length. Oligonucleotide between 20 and 30 to 50 bases are highly sequence specific. So this is why in the start of the reaction, when we talked about requirement for conducting a PCR, we talked about primers, but then we talked about the length. It's because primer length is important. Too much short or too much long primers can result in failure of PCR reaction, either in the form of unspecific binding or not being able to bind if it is too much lengthy. So we have to be very careful in selecting primer length. Like it's just saying, primer length is proportional to annealing efficiency. In general, the longer the primer, the more inefficient the annealing. Now, annealing is very important, as you have seen before, because polymerase chain reaction can proceed through the DNA polymerase can only recruit the free nucleotides if the primer is already annealed. And if it is not annealed, DNA polymerase will not be able to recruit new nucleotides. So the primers should not be too short, a specificity, and they should not be very long, because again it can cause problem in annealing of the primer. Complementary primer sequences. This is very important. It has to be kept in mind that primers need to be designed with absolutely no intra or inter-primer homology beyond 3 base pair to reduce the intra or inter-primer dimerization. Now, to avoid this, dimerization process, this has to be kept in mind, and that primers should not be designed in such a way that they can bind with each other. This can because if this happens, this can reduce the availability of primers for the desired reaction. So this, while designing primers, that it has to be kept in mind. Very important. Now, GC content, as you know, that in DNA strands, in two DNA strands, we have two types of hydrogen bonding. One type of hydrogen bonding is AT, that is double hydrogen bonding, and the other one is GC. GC obviously is more stronger than AT because it is triple hydrogen bonded. So to be able to produce a good quality primer, ideally a primer should have a near random mix of nucleotides at 40 to 50% of GC content. So GC content should be around 50%, because this will help the primer to bind or to anneal with the DNA, because it will result in more stronger or accurate binding with the DNA. there should be no poly G or poly C structure that can promote non-specific annealing. Again, this is a very important point. It has to be kept while you design primer. Through programs like Gene Runner, you have to select one that that doesn't have poly G or poly C stretches, and because these poly G or poly C stretches can possibly create non-specific annealing. This has to be kept in mind. Now, 3' end sequence. The 3' terminal position in PCR primers is essential for the control of mispriming. Inclusion of a G or C residue at the 3' end of primer helps to ensure correct binding. Stronger hydrogen bonding, again, of GC residues here also plays an important role. Melting temperature. As you know that annealing process happens at 54 degrees C. The relationship between annealing temperature and melting temperature is very important. A general rule is to use an annealing temperature that is 5 degrees C lower than the melting temperature. Both of the primers should be designed such that they have similar annealing and melting temperatures. So this is also very important point while designing primer. There are different types of PCR machine. One is the real-time PCR, other one is the quantitative real-time PCR, touchdown PCR, and gradient PCR. These are the different variants of PCR that are used for diverse range of purposes. So it also depends on the type of experiment that you need to run for conducting PCR. Real-time PCR is used to amplify and simultaneously quantify a targeted DNA. It enables both detection and quantification of a specific sequence of DNA. Then we have QRT, a reverse transcription polymerase chain reaction. It is used to amplify a defined piece of RNA instead of DNA. What it does is that when we isolate RNA and then we make cDNA of that, or include the process of reverse transcription, then it can be quantified through quantitative real-time PCR. Now, as you know that the central dogma of molecular biology is from DNA we get RNA, and from that we get protein. So the messenger RNA level tell us about reflect the level of gene expression, information about cell response to different type of stimuli, and protein production. Not always means that sometimes the RNA is not translated to protein, but in a lot of cases we see that the messenger RNA is translated to protein. Here is an example of a real-time PCR. Certain stimuli, in this case is the stimuli is the androgen hormones, and they are removed. This is a gene. This is from one of our published paper. Maybe L09 is increase in expression. The stars represent statistically significant increase, and then it comes down. So this is measured through quantitative real-time PCR, and one can see that these 2, 3, 4, 5, and 6 days after the hormone was removed, the gene express and show a fold change increase that is statistically significant from the day from the control and day 1 and day 7. So this is how the gene expression can be measured or can be studied through quantitative real-time PCR. Now there are numerous applications of PCR. One of the most important is DNA sequence technology. We can sequence DNA through this procedure, isolation of particular gene from genome and DNA, amplification of DNA, like we talked about. Diagnosis purposes, diseases like AIDS, for instance. Mutational analysis, you can compare different sequences and see if any mutation has occurred by comparing the sequences. And quantitation of messenger RNA in cells or tissues, this can be done. Forensic medicine, like crime scene investigation, can be done. And quantitative real-time PCR tells about quantitation of gene expression, drug therapy efficacy, and drug monitoring, viral quantitation, pathogen detection. These are the different purposes. Now I give you an example of how crime scene investigation can be performed with the help of PCR. These are different possible samples that can be collected from any crime scene, including blood, teeth, semen in cases of rape, saliva, hair, urine, bone tissue. So if the crime scene investigator collect any of these evidence, they can possibly reach to the culprit of the crime. So here is a case study where crime scene cadets are studying the crime scene to this cartoon, and they are try to use DNA profiling to determine which suspect cannot be excluded from suspicion. This is the gel which was run after collecting any of the sample that we discussed about. So this is the standard, and now these are allele ladder. This is the one that was collected from the crime scene, the CS. When it was run on the gel, it gives us these two bands. Now, as you know, every crime has suspects. So in this case, we have these four suspects. So one is suspect A, one is suspect B, one is suspect C, and another one is suspect D. Samples are collected from these different suspects, and it is run with the one that is collected from the crime scene, or the CS. So when it is run with the one from the crime scene, and we compare it with the suspects, we can see that the bands of the suspect A doesn't match with the one from the crime scene. The bands of the suspect B are matching with the one from the crime scene. Suspect C are not matching, suspect D are not matching. So it means, it shows that suspect A, C, and D are innocent, while suspect B is the one that cannot be excluded, because the sample that we got from the crime scene and the sample that of B are matching with each other. So it can be concluded that suspect B is the culprit.